everyone can read their own doa. Okay, so for today's lecture for topic 4, we're gonna is about the basic local sequence alignment search, which in general is the blast section. Is the blast bioinformatic tools that you guys have been introduced previously in the previous practicals. So in this lecture, we're just gonna do a bit more in depth of how do we maneuver in the maneuver and use the blast bioinformatic tools. Also, how does exactly, like behind the scene, how does it work and what algorithm is used. So what I'll be talking about today is on the introductions of BLAST, scoring matrices, alignments, algorithm, and statistical significance of pairwise alignments. Okay, so there's 3 learning outcomes for this lecture. Firstly is for you to understand the applications of BLAST as a bioinformatic tool. BLAST is a very general, globally, and widely used among bioinformaticians and biologists. Is a very easy tools to use, and it's almost like a one-stop center if you know how to use it. Secondly is to analyze the various steps and parameters involved in a blast search and the significance of a scores, which is, we call it as value, P, which is P value, and E values. And last but not least is to evaluate the significance of blast query results. So once we get the hit list from the blast search, we need to not identify, we need to interpret and analyze it accordingly. And so the last section will guide you on how to analyze, evaluate, and interpret your results. So what is BLAST? It is a sequence analysis program available out there to allow rapid comparisons of a query, which is your interested sequence, database. Your sequence can either be a DNA nucleotide linear sequence or protein amino acid sequence. It also allows you to utilize a fast, accurate, and web accessible algorithm. And also in the blast searches, it provides you a fundamental understanding of the relatedness of any favorite query sequence, your query sequence basically, to other known protein or DNA sequences. The 6 applications of BLAST is ideal to determine what orthologs and paralogs are known for a particular protein or nucleic acid sequence. You should know the basic of what an orthologs and paralogs are. You can just Google it. It's fairly easy to understand. Secondly is to determine what proteins or genes are present in a particular organisms. If you have a species-specific organisms and you wanted to do matches according to that species, you can use BLAST to do it. Thirdly is to determine the identity of a DNA or a protein sequence. Let's say you have an unknown DNA sequence or an unknown protein sequence, we can use BLAST also to identify what the sequence are and look for the matches and hits. And fourthly is to discover new genes. Let's say if your unknown sequence, be it a DNA or amino acid sequence, have no matches in BLAST, most likely it could be a new genes, which is a de novo gene that has yet been reported in published papers out there. It's actually it's quite impossible for BLAST for not showing any results unless you set the parameter wrongly. But it is possible to find new genes in using BLAST. Fifth is to investigate the express sequence tag EST that may exhibit during alternative splicing, because we know in a translated protein will undergo protein translation modification and called a modified protein. So we can also understand better on the EST tags and the alternative splicing method and process. Lastly is to explore amino acid residues that are important in the functions and or structure of a protein. Likewise, a protein a modified protein will have different structures from the previous translated protein, and the structure will determine the functions of the protein later on. So it's highly important. Let's say if you only have the amino acid residues, so from that amino acid residues, you can actually make a guess of what the function is, because from the amino acid residues, you will know kind of more or less the structure, the possible structure of the amino acid residues, amino acid sequence, and from that structure, you can kind of guess of what's the functions of the protein. And that's why mutated sequence will produce a malfunction protein, because it won't transcribe into the normal RNA, and it won't translate it into a normal protein, and it won't have a normal structure, and that's why by the end of the day we call it as a malfunction protein. Okay, so there's 4 components to blast searches. First, we need to know we need to specify our sequence of interest. Secondly is to select a blast program. There's 5 main blast programs that you can choose, but you need to choose it wisely depending on your goals and aims and objectives. While the third is to choose a database to search. There's a lot of databases out there also. And the fourth is to apply additional parameters. Fourth part, additional parameters are there for you to choose if you have certain certain hits or results in mind. And what I'll be explaining after this helps you to understand why does applying additional parameter is highly important, because if you tweak something in the parameters, you are not using the default parameters, it's not wrong, but you need to know why you are using it, and you need to justify why do you choose that parameters. If you don't have any specific additional parameters needed, you can just use the default setting as it's also as well. So there's no problem with it. So the first step is to specify sequence of interests. I know that you guys probably did this in previous practicals. So we will cut and paste the DNA or protein sequence in the FASTA format, and then paste it in the query sequence sections. You can also use a session number that can be retrieved from the reference sequence or the gene bank identification GI number. So this is the query box sections, while this is the database that you can choose. There's a list of databases here in the drop-down menu button. This is the interest query sections, which provide you additional query alongside with this query in the box. It's like what we did on the Access database practical. We wanted to have results that only matches cyclotide in their title and in Malaysia, is an example. So if you have any other example, if you have any author in mind, you can put the name of the author, but you just have to clarify that that's the author's name. So if you do this, bear in mind that your query results will be lesser because it's becoming more and more specific. And last but not least, at the bottom one, there's an option for algorithm parameters that you can use more. But if you satisfy with what you have right now, you don't have any additional parameters in mind, you can just click on Blast, and the Blast will do your job. Okay, so the second part is to select a Blast program. There's 5 main programs, which is the first one is Blast P, secondly is the Blast N, which is these two are the most common ones out there. The Blast P compares an amino acids query against a protein sequence database. So you put in your amino acid sequence, and then it will produce an amino acid sequence as well. Blast N will compare a nucleotide query against a nucleotide sequence database. The top 3 are a bit more trickier, but you can use it if you know how to use it, and if you know how does it work. So Blast X compares a nucleotide query sequence translated in all reading frames against a protein database. It will translate DNA into 6 potential proteins and compares each to all members of a protein database, while T+N compares a protein query sequence against a nucleotide database dynamically translated in all reading frames, and it denotes whether a DNA database encodes a protein that matches your query. T+X lastly compares the 6-frame translation of a nucleotide query against another 6-frame translation of a nucleotide sequence in the database. It is computationally very, very intensive. The first two are quite relatively easy to understand, while the bottom three are a bit tricky, as I said previously, but there's a summary here. Okay, so Blast P will use protein as the query and do a single search against a protein in the database. That's while Blast N again will use DNA or nucleotide sequence query and then do a single search against a DNA sequence in the database you have chosen as well. Okay, Blast X, T+N, T+X, what they did is if you put in a query, a nucleotide query in Blast X, bear in mind is that in the nucleotide query, there will be a poly A, there will be a poly A tail, there will be a coding region, non-coding region. So what Blast X do for you is that from the DNA sequence, it will only take the coding region that can be translated into reading frames, okay, into reading frames, and then do and all these 6 potential protein will be find it match against a protein in the database. Okay, while T+N, from a protein sequence, it will try to match itself into a DNA punya potential open reading frames available in the database. While T+X use your DNA and then your DNA sequence query and then produce 6 possible amino acid or open reading frames proteins sequence, and then and then search 36 times against a DNA sequences possible operating frame available in the database also. So basically, in brief, what you can kind of see and kind of understand here is that Blast P and Blast C is fairly simple, because only use one single search against one thing and another thing, while Blast X, T+N, T+X is a bit more complicated, and it requires a bit more time for the algorithm to do their calculation and determine how exactly the matches would be. So I'm not saying you guys can't use this, but only use it when you know it. Because personally, I never used those bottom three before. Only use the Blast P and the Blast C in daily basis. Okay, so that's about it. This is the step 3. We need to choose a database to search. If we didn't choose anything, the default setting would choose the non-redundant NR database as our setting. But there's more available databases that we can use as shown in table 4.1. It contains the list of protein sequence databases that can be searched by Blast. There's NR, Mansfield, Sprott Reference Sequence, PDB. PDB is quite popular as well. PAT and Environmental NR. Meanwhile, in table 4.2, it provides you the nucleotide sequence databases that you can use out there, available out there. You can use any that you want. You just have to justify why you choose that databases. Like everything you do, you need justification on why you use it. There's no right or wrong in you just have to know the reasons behind it. Okay, so step 4, last but not least, to apply additional search parameters. There's available parameters such as organisms. If you have If you're working with a very species-specific project like E. coli or bacteria, certain virus, so you can choose which organism database that you wanted it to be. While entries query, like I've said previously, if you have any additional parameter or query that you wanted to include in, like a name of an author, a place, a name of a country, and more, you can put it in here. And last but not least is the algorithm, which is the Blast P, PSI Blast, PHI Blast, and more. It depends on what kind of Blast program you are using. PSI Blast, PHI Blast, you guys will be having these practicals on this kind of these two Blasts in practical advanced Blast. I believe nets practical. So yeah, so this is an extra example in Blast N and Blast P. So in Blast N, we can choose an aspect size, which is aspect size, which is the E value, the word size, the match or mismatch scores, also some filters if you want. Similarly, in Blast P, you can choose the aspect size, word size, scoring matrices, like Blossom 62, Pam, Blossom 80. You guys have heard a few of these names previously in previous practicals. And filters. Yeah, guys. Another 10 minutes benda ni padam. Lepas tu nanti join balik macam biasa. Thank you. About this G plus loan, I haven't yet have the time to ask Dr. Fiza. So I don't want to say much about it because I don't really have much knowledge regarding to say about the gap course. So if you guys want to know more about it, maybe you can ask Dr. Fiza or maybe you can Google it. Okay, so that's just basically it. And other optional parameters are included in the post search. So although it's important to set everything correctly before you clicking the Blast button, but you can do few other parameters changes post search as well. You can choose the organism to search, turn the filter on or off, change the substitution matrix programs, change the aspect E value, change the word size, and also change the. So this is example why exactly choosing the settings or choosing the setting is highly important for your result sections. This is because let's say I'm going to show you 4 examples. Okay, this is example 1A. If you take the human insulin as the query using the Blast PA as a program and selecting the C. elegans reference sequence as the database with the default setting to unfiltered. So as you can see here, the aspect value is 0.034 with an identity of 29%, positives of 40%, and gaps of 13%. So this is when you search human insulin against worm reference sequence protein by Blast P using a default parameters, you get these results. Okay, for the second example B, still using the same query, the same program, the same database, but we change the option into a no compositional adjustment. You'll be seeing that the bit score, aspect value, and percent identity all change with as compared to the previous settings. See here, previously it's 0.034, but now it's 0.009, while the identity 40%, 41%, and 14%. Okay, and then another example, if you're still doing the same query, program, and database, but now you change the option into a conditional compositional score method adjustment. Again, the scores, the aspect value, the identity, positives, and gaps value also changes. And again, for the fourth example, if you're still using the same query, program, and database, but now we'll choose the filter low complexity regions as our options. So the scores, aspect, identity, and positive value are all change. They change much, and the query result sections also. Although maneuvering along the Blast programs are fairly easy because everything's there. You just have to choose whichever you want. But when you choose it, it's highly important to know why exactly you're choosing it also, because and if you have more than one sequence to evaluate or identify or studies, it's better to have a very standardized settings for parameter setting for all your sequences. So okay, I'll show you the blast search output page once you're done with all the settings and you satisfied with your setting. You click on the the blast button. It will do its job for a few minutes, and then it will provide you this output page, which contains you the query, um, summary, the database that you have chosen, the program that you choose and currently running on, and also other reports such as the taxonomy reports, distant tree of result, related structure, and multiple alignments as well. Bear in mind, this look oddly not familiar because this is the previous looks. The Blast program already changed their look to what you used previously, but most of the stuff are relatively similar. Okay, so why I said previously that Blast bioinformatic tools is, I kind of say that it's a one-stop center because it provides you more than just alignments results. So it's very very helpful, and you can gain a lot of information from just one program. Okay, so in the taxonomy report, it will summarize the species with matches. So if you have your sequence, and it will and click on the taxonomy report summary, it will provide you a list of the summary of relatedness species between with highest matches. Let's say here 18 hits as compared to other hits. So your sequence, this example in this lecture sequence is closely related to a Homo sapiens. It's either closely related or it is a Homo sapiens sequence. Okay, so if you have 15 examples, if you have 15 result hits, so most probably you'll get 15 lines as well. Okay, and then the blast search output would produce a tabular output that provide you the identifier for the database sequences, the description of the annotated names, the score, which we call the S value, and the EU value here. What you can see here is that a high scores will have a low E value. That's actually what we want, and I'll explain more on why we want a high S score with a low E value. And you know that you can download the this section here by clicking the download option and choose FASTA file or CSV or Excel and XML. Okay, and then the blast search output also provide you a graphical output. Let's say your sequence query has a 17 hit result, so there will be 17 lines here provided that will align accordingly to which it is most similar along your query. And the best thing is that it's color coded accordingly to the alignment score, so you can make your own judgment and interpretation by just one glance, and it will save your time. So it's fairly good. And let's say if you have you have 17 matches hits, but you have two that you are keen to know more about it, so you can choose your query by clicking the box here and then choose the multiple sequence alignment options, and you can simultaneously see the sequence alignment in more than two sequences. Okay, so basically the previous sections are the one that you need to understand on how to use Blast program in general and how do you maneuver along the sections. So now in these sections, we will talk more on how does the original Blast algorithm works. which have three phases. This is highly important because before you choose your settings, parameters, algorithms, you need to know why you are choosing it, why you are changing it from the default parameters, and because just in case if people ask you why do you choose value of other than the default parameters, and you will have your own reasons. Okay, so there will be three phases in the Blast algorithms. So the phase one is a compilation of a list of word pairs. We call this the word size equals to 3. I mentioned previously in the algorithm parameters options, right? In the Blast and in the Blast P sections, there's word size that we can choose. So word pairs, we call it word size in a certain threshold called T. So as example, for a human RBP query, the FSG the query was in yellow. I just have to say that I don't know why there's the yellow part there. I couldn't see any yellow. I can only see red stuff. And I haven't talked to Dr. Fiza yet, so let's just discard that. So how does the word size tree works in the algorithm? So the way is, as you can see here, this is an amino acid sequence of the human RBP. It contains the FS. So as what you can see here, this is an example from this query sequence only. So FSG is the first three sequence, and then continues on into SGT, and then GTW, and then TWY, and then YWA. So it goes like that. Only if you have the word size of 3, but you have the word size of 4, let's say 4, 10, 11, you can choose whichever you want. It will be, let's say we're gonna use word size of 4. So the algorithm, the BLAST algorithm in phase 1 will read it as FSGT, SGTW, GTWY, TWYB, things like that. So the word size is actually important because you are setting or telling the algorithm how to read your sequence, and it will actually impact and affect how the results, your result by the end of the day. So okay, this is the word size. So basically from these slides, the most important bit is how does exactly the algorithm in phase 1 read the read your sequence. Read your sequence. It is in the form of a certain word size. Okay. Okay. So um, can you imagine like a long DNA or amino acid sequence, and if you determine, if you're telling the BLAST algorithm to use the word size of 3, so it will read the first 3 and then the next 3, the next 3, the next 3, the next 3, up to a point there will be an overlap. It's not an overlapping, up to a point there will be a similar pattern in that have been identified previously, so it will identify which exactly it is being positioned, and that's why there's this number, the 6, 5, 11. These are all the positions of where exactly the GTW has been found along these sequences. You don't need to like really understand how this happen. You can, I would suggest you to find like a short YouTube that tells you about the BLAST algorithm, or how does word size BLAST works. Um, the only important bits is that the threshold level, we can also identify the threshold level, in which here we set as, and default, in the default setting it is set as 11 normally. You can change if you want, but if you're not changing anything, the threshold level will always be 11, and then only neighborhood words or hits with more than 11 will be retrieved, while neighborhood word hits with low than the threshold level, basically 11, will be discarded. So that's how in the phase 1 the word size help the algorithm, no, the algorithm in phase 1 helps the search or query searching to eliminate few results, because there's a lot of results out there, right, in the database. So how does exactly the algorithm retrieve and eliminate few available sequences? Okay. Secondly is the pairwise alignment scores, which are used to determine using a scoring matrix. The pairwise alignment scores are generally called, also known as the substitutional amino acid scoring matrices. That's Blossom 62, Blossom 80, PAM as more. What they do is quite different from how does the word size and threshold level work. The word size and threshold level work together as one, while the pairwise alignment scoring matrix works on its own, in which it is a program, or it is a program that will count or will determine the scoring if there's a base substitutional between your query and the possible sequence, your possible hit sequence. Let's say example, if the supposed V in your 24th residues are changed into N. So at the 24th residues, it is counted as -3, and then the algorithm will calculate from the first residue to the last residue, and will calculate everything, and only the residues at a certain cutoff level will be retrieved for the S matches, while residues in a certain cutoff level that while sequence below the cutoff level will be eliminated and discarded. So the phase one are more likely to filter most of the stuffs during pairwise alignment searching. So the first method, the word size and the threshold level involve the neighborhood hits, while the pairwise alignments involve the substitutional based scoring matrices. Moving on into phase two, the phase two are quite easy. Scan the database for entries that matches the compiled list, and this is fast and relatively easy because there's no such calculation here. They only try to find and match here and there, while the first phase, that they all the counting, calculating and determining the cut level and threshold level and more. So moving on into the phase three, the last phase, when you manage to find a hit, which is an example, there's a match between the word and a database entry, it will extend the hit in either directions. Like an example provided here, if let's say the RBP is your query and the lactoglobulin amino acid sequence are the hits, the algorithm will try to find the hit that they share and then try to extend it, although it doesn't have the same sequence lah. So in phase three, it will keep track of the scores using a scoring matrix, and that will stop when the scores drop below some cutoff level. The cutoff level too, you can actually set it. Okay, the cutoff level is the T level, word size, probably E level, P value also. Okay, so this is just an example. They will firstly identify the hit, and then they try to extend it, and we call that as extensions. Okay, so what is threshold value? I previously mentioned the default threshold value are 11, but for blast and the word size, for blast and the word size is typically 7, 11 or 15 for an exact match. Remember kan, the word size to the first three, contoh if let's say the word size is 7, so the first 7 sequence and the next 7 sequence, next 7, okay, changing the word size from 7, 11, 15 or as small as 3 is likely to change the threshold level of a protein. Okay, let's say if the word size is 15, it will give fewer matches and is faster than word size of 11 or word size of 7. Specifying a smaller word size will induce a slower, however, a more accurate search. You can imagine if you have a very, very long cloth, you're using 1 meter ruler and a 10 centimeter ruler. Obviously, the 1 meter ruler would use smaller time as compared if you're using a 10 centimeter ruler, right? So it goes like that. The analogy is similar in what happening in the algorithm of Blast. So if you have time, you really want a accurate search, you can have a smaller word size, but it will take some time. If you don't mind it to be not that accurate, you can use a bigger word size. Okay, for Mega Blast, I don't know if you guys heard this before, but the word size is normally 28 and can be adjusted up to 64. What it will do is that it is very fast for finding closely related DNA sequences. Okay, normally in published papers or journals, bioinformaticians or biologists will use Mega Plus if they are dealing with environmental samples that is not definitely not species specific. There's a lot of species there, there's bacteria community there, or they are dealing with metagenomics or transcriptomics of more than one bacteria. So most of the time they use Mega Plus because a bigger word size will provide a faster search, but using Mega Blast, it will not just produce a faster search but also a relatively more accurate search. Okay, so I mentioned previously about what is hit and extension, right? So between your query and a DNA and a sequence and a possible hit sequence, the algorithm will try to find the hit region and then they will try to extend it. The hit region are the one that shares the same amino acid sequence or nucleotide sequence lah. And then the rest, they will try to extend it and calculate the scoring matrices. If it's above the if it's above the cutoff value, it will retrieve. If it's below the cutoff value, it will be eliminated. So how does exactly the threshold value affect your search? Okay, the red line are the extensions while the black line are the hits. As you can imagine here, when the threshold value increases from, let's say, the normal setting, the default setting are 11, we change it to 21 here, 21. The number of extension will reduce and the number of hits also reduces. By means, it will be more and more accurate lah, because you will have a more accurate search due to the threshold value. If the threshold value is very, very high, it will only provide you the most it's not a perfect match, but the most match, the match yang very, very accurate for your searching. Okay. And then going into statistic and e-value, you guys heard previously of what p-value is. E-value is relatively similar to what p-value is using. So the aspect value here, as you can see, it is summarized as aspect, but sometimes people call it as e-value. So the aspect value, which is the e-value, is the number of alignment with scores greater than or equal to score S, so that are expected to occur by chance in the database search. This is just the operational definitions out there. You don't really need to understand much if you have any kind of not understand, if you guys have any simpler terms and simpler definitions, it's better. So an E value is related to a probability value P. This is the P value. So these equations below is derived from descriptions of the extreme value distribution. S is for the score value, E is the E value, which contains the number of high scoring segment pairs expected to occur with a score of at least S, and MN, the length of two sequence, lambda and K, which is involved in the Kalin-Askew statistic. I won't be talking much about this because I've seen this a lot of time, but I never ever use it because we're not a robot. We just need to know how to use the e-value provided. Okay, so this is, but you do need to know that this is the formula used to calculate the e-value inside the algorithm of Blast. Okay, so example, this is the score, which is the S value, the bit score, and this is the aspect value, which is the E value. The score is 32.7 bits, while the aspect value is 0.034. Number of alignments, which is HSP, with the bits, which is the E value itulah, with the bit score of 32.7 or higher, is expected to by chance in a 3.4 in 100 times. Sebab 0.034 is equal to 3.4 times 10 negative 1 times 2. Okay, negative 2. Okay, so it's relatively similar to how statistician would explain, but as long as you guys have like the basic knowledge of what p-value is, it works relatively the same of how e-value works. Okay, how does E value is significance? The value of E decreases exponentially with an increasing S, which is the score lah. higher S value correspond to a better alignment. So a very high scores corresponds to a very low E value. That's what we've seen previously in the previous lecture slide. A high S value, a high score will have a low E value. Okay. For an E value of 1, as an example, for an E value of 1 match with a similar score is expected to occur by chance. For a very much larger or smaller database, you expect it to vary accordingly. How do we interpret the BLAST results of E value and P values? Why does in previous statement, it is said that E value correlates with the P value. A P value is a probability of chance alignment occurring with the score in questions or better. Okay, ni another operational definitions. So the relation of E value to P value is as follow, which is this formula. P equals to 1 minus exponential of negative E value. A very small E values are very similar to P values. Okay, that's what we want to know and what we want to remember. The E values of about 1 to 10 are far more easier to interpret than corresponding P values. As you can see here, this is the E value from 10 to 0.0001 and the P value of close than 1 to 0.0001. Okay, a P value below than 0.05, okay, below than 0.05 is traditionally used to define statistical significance. The P value of 0.05 suggested that some observation is likely to have occurred by chance 1 times in 20. So in general, let's say you have 20 20 data, 1 over 20 or 1 over 20 of the data is expected to occur by chance, but if more than 1 over 20 that occurred by chance, you will have more than the P value will probably be more than 0.05, and that's why P value of 0.05 is considered as statistically significant. We don't want random chances. We want things to happen because it happens. Okay, that's why statistical significance are there. So an E value, okay, a P value of 0.05 is said to be statistically significant, and it correspond E value is 0.05. So in general, in brief, we can say that an E value, expected value of below than 0.05 is statistically significant enough for you to have confidence in your results. Good. So this part, the E value, P value, the bit score, we're going to the statistical part of BLAST. You have the results, now you need to interpret your BLAST, whether you have confidence in it, whether it is okay to use in your report, or whether you need to discard it. That's why E value and P value interpretation is very important. So in this section, we'll talk about the row scores and bit scores, which is the row scores. It is calculated from metrics and get penalties parameter that have been chosen during the default settings or algorithm parameter settings. And then the bit score, which is the S prime, is calculated from the row scores, which is from the substitution matrix and get penalty by normalizing with the statistical variables that define a given scoring system. And this is the what you call it, formula. The importance of bit scores. The bit scores account for the scoring system that was used and describe the information content inherent in the pairwise alignment. It allows scores to be compared between different databases, even if different scoring methods are employed or performed. Bit scores can indicate the E value as well. Previously, we used the formula for E value is quite different from this one. If the search space M and N is known, we can know the E value as well. I won't be talking much or more about this slide because it's too technical and it's too statistical wise based and focused. So we'll move on. Okay, so by the end of the BLAST search, we will get a search summary which contains the search parameters, the type of database, the statistic, and the result statistics of our summary. So the search parameters here, we have BLAST P as a program. If we use and we use word size of 3 and aspect value of 10. It's quite high. normally people would choose probably below 0.05, and the hit list size, gap penalties, I didn't talk about it because I don't know much, and metrics, the type of metrics, and also the threshold level, which is 11. In here it summarizes your database, that you have chosen, which number of letters is the length of database, and here are the EVD parameters, which is the Kalin S curve, the one that the formula that have the lambda and k previously. So if you want to manually calculate the e-value, you can do so, and then the result statistic that provides you the m value, n value, and the m times n value. From the m times n, you can find the effective search space. Okay, again, I won't talk much about this. If you have certain questions regarding the technical issues or stuff in the BLAST algorithm, you can ask Dr. Fiza for more clarification. Okay, so in BLAST searches, how to evaluate the significance of your results. So how is firstly to look at the e-value or the expect value, because it is a better indication. Most of the time it's a better indication than percentage identity or a percentage similarity. Don't be surprised is your e-value is statistically significant or lower than 0.05, but with an identity of probably 20% or similarity of 15%. It's possible to happen. So, and then we also need, you can also produce an inference for our homology from the sequence analysis and the protein structure and functions. So you can find more information about the homology between your query and the hits results if you analyze it properly. If you analyze it properly, the sequence analysis and the protein structure. Last but not least, from the BLAST searches, you can also understand better the divergent evolution that is resulting in the number of false negative matches during database searches and the pairwise protein alignments. As an example, carriers of hydrophobic ligand RBP4 and adherent binding protein will exhibit to share very little sequence identity percentage, but closely related in the form of 3D structure. How is this possible? It is actually possible because bear in mind that RNA contains codon, three sequence, right? Codons, and codons have a degeneration property which allows more than one codon to translate it into the same amino acids. So although the DNA sequence and RNA sequence are different, but as long as they have the same protein structure, the same protein structure, they can share the same 3D structure and function. Okay, so moving on, we're gonna talk about more stuff regarding BLAST search. Most probably macam issues or problems in BLAST. So sometimes a real match has an e-value of more than 1. Is it possible? It is possible. Okay, as I've mentioned previously, a high bit score will normally have an e-value lower than 0.05, which is a good thing. And then a similar e-value, however, a similar e-value which is relatively still statistically significant, still lower than 0.05, but however with the very low bits. Is it still possible? It is still possible. And let's say, just let the complement component 8 is a real match. Can we say that it's not a real match because it doesn't have an e-value lower than 0.05 and the bit score is relatively low as compared to the highest bit score? Can we just discard it as a not match? We normally people won't just discard it as soon as possible. So the important thing and the way is to do a reciprocal blast to confirm your finding. What I mean by reciprocal blast is that you have an X query sequence, and then once you blast it, Y sequence Y sequence are provided from in the output page results. Now what you want to do, if you want to confirm whether those two are matches, those two match in whichever form, use the same settings, but now you change it. Choose, rather than using X as the query, now you use Y as the query, and then do expect X to be there in the list. If it says in the list, it's a real match actually. If it's not in the list, then maybe you can discard it accordingly to your judgment lah. So sometimes a real match has an e-value of more than 1. Don't just discard any e-value lower than 0. Any e-value more than 0.05. Macam straight away discard all the e-value more than 0.05. Because sometimes it's possible for the e-value to not be statistically significant, but it is the real match. So how do we assess whether proteins are homologous to one another? Okay. Ni example from previous real match lah. Okay. After we did the reciprocal blast, okay, let's say we are doing the complement component 8 against our retinol binding protein 4 as the query and the sequence and the hit sequence. Although it doesn't provide you an e-value of lower than 0.05 and a bit score of with the math scores as compared to the 414 scores as the most apart. Once you try to do a reciprocal blast, it is still there. So we can say that it's homologous to one another, although it doesn't share any, it doesn't have a very strong identity. So, example, contoh macam ni, the RB4 and complement RBP4 and the component 8 protein, although it have a very low bit scores. Lepas ni jom balik okay. Although it have a relatively a low bit scores and the e-value of 0.27, definitely not statistical significance in our eyes, in our understanding, and with a percentage identity of 25 here. Okay, however, the 25% identity is still in the twilight zone, and if you Google out the twilight zone homology protein detection, there will be a very short reading on what twilight zone range is. Twilight zones are considered as a range from 0 to 100, in which at 25 or 20% level range, it's still considered as confidence enough, although it's not more than 50%. So you can't exactly blame, you can't exactly say that, oh, it's 25% identity, I'm not going to use it. Additionally, is with an e-value of 0.27. I personally think it's not good enough to be used in my report. But if you look closely and interpret it very, very wisely, you can still use it, because normally this kind of protein are called homologous proteins, like here. So, but they are indeed homologous. So, try to do a blast search with complement component 8 protein as a query and find many other lipoproteins. Why do I say it's homologous? Because most homologous proteins share the same, they share the same motif and domain family, most probably. So, they are homologous to one another. So if we take the complement component 8 as the query and try to find other lipokylins, so we see here the one box in the very light blue color here. There's 3 results boxed: the alpha-1 microglobulin biochemical precursor, the complex forming glycoprotein, and the chain E, the structure of human retinol binding protein. These 3 are actually found in the result section when RBP4 is the query. So that's how you do the reciprocal blast. You look for identical matches when you do both sides of blast searching. You change the query into sequence and then the hit sequence into query, and if you find the same exact information listed, most probably those are the real match. But now you just have to wisely interpret it and analyze it why exactly it's still there after reciprocal blasting. Okay, so is the aspect value significant? It's not significant at all because it's from distant matches. So um, in whatever settings, distant matches would not have an aspect value of lower than 0.05 because it doesn't share most of the stuff. It doesn't share the similarities and identity very well. Are the two protein approximately the same size? Normally, but not necessary. It depends on the protein size tu juga. Okay, do they share a common motif? Yes, the one that have been boxed in green ni lah, which is the common motif. And that's why we say those proteins are homologous to one another. Common motif, yes. Can the two protein multiply aligned? Yes, they are aligned to one another. Although they are barely holding at the GTW common motif. Okay, do the proteins share a similar biological protein? We expected it to be yes because they share the same common proteins or domain proteins, so they will act similarly. So do the proteins have a similar 3D structure? Yes, even with diversity in sequences. Okay. Do reciprocal blasts detect the same protein? Like I mentioned previously, there's this three sequence hits here, and it is found in the previous hit list of when RBP4 is used as the query. So yes, the reciprocal blast does detect the same protein. So that's how you do and kind of understand and interpret your sequence, probably not sequence result, your hit results. You don't exactly discard it out of nowhere without no reasons. Just give other sequence a chance and try to understand more. Sometimes that is the real match. Um, so a second issue, the first issue is that E value sometimes is more than one. Okay, a real match has an E value of more than one. Okay, the second is that sometimes a similar E value occurs for a short exact match and a long less exact match. By means similar E values, that E value that have that is lower than 0.05 lah. Both E values are lower than 0.05. This is 2 E negative 13 for E negative 14 negative 0.8. Okay, however, this one has an identity of 94%, while the bottom one has an identity of 31%. Although both E values is statistically significant, but the identity percentage differs between one and another. Is that okay? That's actually okay as well. Right now, you just have to look why exactly it doesn't why exactly the percentage identity turns out to be 94%, why does it turns out to be 31%. There's no right or wrong, you just have to know why and how it works. And this is actually this kind of issue such as E value more than one or similar E value but percentage identity varies are possible to happen because it all depends on the algorithm of the blast that I previously stated, yeah, the phase 1, phase 2 and phase 3. So actually, although this kind of stuff is fairly technical, and I said previously that we're not robots, we're not calculators, we don't need to calculate all the E values and stuff, but we actually need to know why and how the aspect values are that, why the percentage identities value are reported like that. Because from there, if you understand the algorithm, all the phase and all the algorithms used and the performance, you can actually make your own judgment why the identities are such as 94%, why the second identities are such as 31%. So it's fairly good for you to understand how the algorithm works actually. Is everything okay so far? ke? I hope it's okay. So moving on. So in blast searches, okay, this is another issue. If you have too many results in your result page, so what you can do is that you put in your query a search in the reference sequence database and reduce, because it will reduce the redundant database matches. It will provide you a smaller list lah. And then also to limit query to organism-based database. If you're working on with E. coli, just set the database to E. coli and it will only provide you E. coli related sequence rather than the overall sequence macam tiba-tiba Homo sapiens and such. So you can select a taxonomy identifier TXID of interest through the options features in the Blast server. You can search it and then limit the search by organisms or do limit search to specific organism before the query. Also remember that you can do pre-search and post-search optional parameters setting also. Okay, and then thirdly, you can use only a portion of the query sequence when appropriate. As example, a search of multi-domain protein can be performed with just the isolated domain sequence. Let's say, you know that a nucleotide sequence contains all the nucleotides of the genome of that sequence lah. It will contain coding region, non-coding regions, and the poly A tails. So if you're trying to find similar informations, if you're trying to find similar proteins with the same protein domain functions or protein domain motifs, so you can actually just highlighted the domain sequence and just put in the query sections and it will only provide you informations that matches to the domain sequence in your query. So it actually provide you and smaller list. But only do this if you know that that sequence you're using is the isolated domain sequence. Because sometimes it's not when you think it is. So it's okay, boleh lah. I'm gonna start. Okay, dah tekan record. So as I said previously, you can use isolated domain sequence and put in your query if you wanted your result list to be smaller and shorter, but only use this kind of performance or way method only if you know that the sequence is the isolated domain sequence. If you 50-50 whether it is true, whether it is not, don't use it. But you can always have further readings in published papers. Most probably there will be reported sequence of what your interested domain sequence are. And then you can also adjust the scoring matrix and again adjust the aspect value like I said previously. If you really want only the statistically significant results, you can change and set the aspect value to 0.05. But if you're okay with at least 10, because the p-value will still be relatively low than 1, so you can change you can set the aspect value lah. If you want less result, aspect value tu tinggikan. Increase the aspect value settings during the parameters. Okay, and then if there's too few results in your blast search, what I can recommend is to remove the entrance limits rather than putting search that only hit that sequences hit that matches with your query and your interest query, such as like probably specific to Malaysia or have a name of a certain author. um, Remove those query and just provide the algorithm to give you more freedom and more options. And then secondly is to raise the aspect value. um, Okay, and then thirdly is to try scoring matrices with higher PAM or lower Bsm values. And then last but not least, to search all available database instead of specific database like I mentioned previously and shown in table 4.1 of table 4.2. For protein sequence, there's more than 5 up to 10 respective protein sequence database, while the nucleotide sequence database has more like 10 to 40 specific database that you can choose from the NR database, PDB database. You can do more than one available database. You can search in one more than one available database lah. Cuma the only thing is you need to find it one by one. That's just like the cons, but it's not really a cons because it's there. You just need to choose in the drop down menu button lah. Okay. So that's it. If you have any question, feel free to do so. Try to answer my best. I know this is fairly very technical. I said technical many times before, but it is a very technical lectures. I know that it's I just hope that it makes sense. Although it's you might forget it by the next 2 or 3 days, but it's okay. This lecture will always be here. There's available summary or